and be able to see. So each of those are representing, galaxies within our universe. Our speaker, Dr. Juna Kollmeier, who will join us in a little bit later, she is the next director of the next Sloan Digital Sky Survey. And so we are talking with her about how we can get data from the next digital sky survey into open space. One of the things that Eric mentioned before was about a project with Michelangelo's David statue. And this was a definite departure for us in terms of visualization, but in some ways, but not in another, because this is really about very large-scale data. And so this, several years ago, Mark LeBoy from Stanford University somehow convinced the folks at the Academia Museum in Florence to give him full access to Michelangelo's David statue. And he used a high-resolution laser range-finding scanner to scan the statue at sub-millimeter resolution. And then he needed to visualize all of those millions of triangles. And that's where we came in, because we were able to do it. And so we're zooming in here, and you can start to see the little triangles that are there in Michelangelo's statue in his face. We could also change the lighting, so when the real statue was outside, you could see what it looked like at different times of the day. So we thought that was pretty cool. But what is even cooler is we now have this working at much, much higher resolution. So these are my colleagues, Valerio Pascucci and Chuck Hansen, who are doing twofold things here. So what you're seeing now is almost 1 billion triangles, little triangles there, and each one of these frames is 2 gigapixels in size, not megapixels like your phone, but gigapixels. We're going to zoom into that same place in the eye that you just saw in the previous one, and at this resolution you can't really see any triangles. So it is a near perfect digital copy of Michelangelo's David statue. And one of the things that we found out about the statue when we were looking at this is that when the statue was outside, hundreds of years ago, someone, I'm thinking maybe like a teenage boy, carved their initials into the back of one of his legs. And what you will see in this next rotation here, and when we zoom in, are the initials that were carved into the back of the leg of Michelangelo's statue. I don't think you can quite get close enough to it now in the museum to be able to look at that. So it's an amazing. Here we go. We'll see if we can see. Can you see the initials N over here? Who is N? We don't know. It's a historical mystery. We also have an application that you can use to be able to look at this on your iPhone or iPad. All right, so back to science and big data. This is a really exciting international project called the Connectome Project, and it's a set of neuroscientists throughout the world who are using high-resolution electron microscopy and other microscopies to try to get at the fundamental wiring diagram of our brains. This is the way that they do this is also remarkable technology. They take the highest resolution electron microscope and they aim at the smallest possible piece of the brain, and then they save the data, dump the data, and then they go to the next one. They go, it's like image, save, dump, image, save, dump, image, save, dump, and these things run 24 hours a day for 7 days a week. When all the data comes out, it doesn't quite look like this, so we have to have algorithms to put it together. You see that little red square that's in the upper left there? That's the maximum size of an electron microscope, and that's basically 4,096 pixels by 4,096 pixels, so 4K. So if you're lucky enough to have a 4K projector or monitor at home, that's that size. And then what they do is they do the next slice, and the next slice, and the next slice. We get this exquisite resolution in the plane, but they can't do that same process when they do the depth. So you'll see that things actually change quite a bit as we go, even though they're cutting the smallest that they possibly can to get there. Two things to note about this is that the overall resolution that they are able to get is 2 nanometers per pixel. That's 2 times 10 to the minus 9 meters per pixel. in terms of resolution. And one of those little bitty data sets is 16.5 terabytes of data. So not your megabytes or gigabytes, or gigabyte, it's the next one up, terabytes. So we have developed technology, visualization technology that enables them to interactively visualize those entire 16.5 terabyte data sets. And so there is one of the connectomes on our wall, and the neuroscientists were thrilled. They spent days and days looking at this data because no one had ever seen the brain at this resolution in this particular area. So it was like Leeuwenhoek with a new microscope. And really, this is why we think of this as a new microscope. Robert Mark and Brian Jones, the neuroscientists who created this new technology for creating this data, and then we were able to take it and visualize it, and they were able to see things that they had never seen before. They made scientific discoveries that they had never been able to make before because they could see at these higher resolutions. So it was really exciting. And this is an animation of just one cell type in the connectome, in this particular connectome. And when people have told me our brains are highly connected, and we all have a mental model of that, and then when you see that, remember this is at 2 nanometers per pixel, how small a piece of brain that is, and this is only one cell type. And when they put in all the cell types, there's no space that you can see there. It gives you a real different sense of what it means for the connectivity and the complexity of those connections within our brains. So staying with biology, but switching to different types of large-scale data, I want to tell you about work that one of my colleagues, Mariah Meyer, is doing for visualization in biological data. And Mariah is doing something which we call information visualization, where instead of 3D and spatial plus time, she really is dealing with higher dimensions of data and not the 3D spatial. So you can think of things like social networks or financial data or genetics or things like this. She's been working with a number of different biologists in which she goes in and interviews the scientists, reads their papers, find out what their main scientific questions are, looks at the tools and the visualization systems that they're using currently, which are kind of usually these kind of one-size-fits-all, and then finding out what works and what doesn't. And then she goes away and designs from the ground up a new visual analysis system to solve their particular biological problem. Puts it in their hands, the researchers' hands, gets feedback, does another version, gets more feedback, does another version in this rapid prototyping. And here are 4 of the systems that she's created. And you'll see that they all look a little different. And that's because they're solving 4 different biological problems instead of just the one-size-fits-all. So let me tell you a little story about one of my favorites. This is a system she has created, which is called MISV. And it was done with researchers at the Broad Institute, which is a combination between MIT and Harvard, some comparative genomicists who were comparing the chromosomes of humans, which are represented in the outer ring, with the chromosomes of other mammalian species. And they had a model for how these chromosomes should map. And this is the first time they looked at their data in Mariah's system. You'll see those dark red and green bands at the top there. Those are correct. All the other ones are wrong. And they immediately knew that their model was wrong. And that's another great thing about visualization. And because we're so visually oriented, when we see something, we often know it's like immediately, wow, that's wrong. So visualization is good for debugging in many ways. So they spent a lot of time going back and working on their model. And here's an intermediate version of their model. And you can see it's better than the first version. But at this point, they understood, they decided that they had something fundamentally wrong with their model and they needed to start over from scratch. And they used Mariah's visualization tool and their own theories and data to come up with a new model. And here is the solution, the model they came up with, that was experimentally verified and then published. Mariah asked the principal investigator of the research, how long do you think it would have taken you to get here without the visualization tool? And he said, quite frankly, I don't think we ever would have made it. And this is really the power both of the right visualization tool, but also of collaborative science, of two people or groups that neither of them could have solved this problem by themselves, but only working together in an interdisciplinary way that they could solve this important problem. And I do want to give you the sense that you saw some stills here, that interaction is so key, and it's so key to our thinking of hypothesis testing, of try this, get feedback, try that, get more feedback. And we want to, you really have to have this kind of interactive feedback. And so this is kind of a sped up little version of the kinds of things that they were doing and bringing in different data sets and different tools and different types of things. Mariah continues to be very popular among scientists and wanting her to help them with their science. Let me tell you another great biological story about a collaborator. This was a collaborator who was a developmental biologist, Chi-Ben Chen, who was looking at how zebrafish develop. That was his model organism, and he developed a transparent zebrafish so he could watch how the organs were developing. And so they put video cameras in there, watching over time, every day, every minute, of how the zebrafish was developing internal organs. And then the poor graduate students had to take those videos and then like segment out all the different organs and over time, and it would take them weeks and sometimes months. And so the tools were just not there for doing it. And Chi-Ben Chen came to us and said there had to be a better way. And my colleague, Dr. Chuck Hansen, said, yeah, I think we can work on this. And so over multiple years of time, they created this system that has grown in popularity with biologists who are using confocal microscopy and other imaging modalities to be able to do these multi-channel, very complicated systems to track cells, to be able to do that segmentation of developing organisms, as well as very large scale systems. And let me play this little video that talks a little bit about flow render, but also one of our collaborators using it.
Flow render is a tool that was developed for confocal microscopy data, and it's focused on giving biologists an easy-to-use method for doing visualization and analysis of 3D confocal microscopy data. So Gabrielle Carnegie here at University of Utah has found that flow render is a valuable tool for her research in the Carnegie lab.
The Carnegie lab works on congenital defects in muscle development, such as congenital diaphragmatic hernias. We're using it to image developing limbs and developing diaphragms, which are very complex three-dimensional structures. So I would say that we would be unable to do any of our work unless we had flow render.
That's what you want to hear from the people that you help, that you're making a difference with it. It also creates these amazing, beautiful images, and I know that people have used this. There are these biological image photography and image competitions internationally that Nikon and other groups sponsor, and people have used flow render to win some of those competitions. It creates these amazing systems. So let me tell you a little bit about some of the research that I'm doing and my colleagues in uncertainty visualization. One of the things that is happening is that as visualization is being used more and more for understanding data, we have to understand that none of that data is perfect. It all has errors and uncertainties associated with it. And so one of our real challenges is how do we go beyond the showing of the data and finding those patterns, but also enabling us to understand the levels of uncertainty and error so that we can make better decisions under uncertainty, which is what all of us do every day anyway, but especially when we have scientific data, whether it be experimental or sensors or computational, it all has a relative amount of error. So what are ways in which we can do this? That top visualization in the middle there is work done by two of my colleagues at the Ski Institute, Ross Whitaker and Mike Kirby, and they came up with a way, oh, that's not good. Don't you love it?